this video, we'll talk about the concept gluconeogenesis. In the word gluconeogenesis, gluco means glucose, neo means new, genesis means synthesis. So in gluconeogenesis, there is a synthesis of a new glucose, or there is a production of new glucose specifically from non-carbohydrate precursor. What do you mean by non-carbohydrate precursor? Non-carbohydrate means apart from the carbohydrate. So in gluconeogenesis, there is a synthesis or production of new glucose apart from the carbohydrate precursor or carbohydrate source, like lactate, pyruvate, glycerol. Even from some of the amino acid, new glucose is synthesized in gluconeogenesis. When there will be synthesis of new glucose? Obviously, when there is a less glucose level in the blood or in the body, or when there is a low blood glucose level, this gluconeogenesis comes into picture. So when there will be less or low glucose level in the blood, obviously during the prolonged starvation or even after vigorous exercise, or when there is a long gap between the meal, or even during the overnight sleep, there will be less glucose level in the blood, during which gluconeogenesis comes into picture. Where it will take place, 90% of the gluconeogenesis or most of the reaction or most of the gluconeogenesis takes place in the liver, and some amount of gluconeogenesis of about 10% even takes place in the kidney, that is cortex region of the kidney. So most of the gluconeogenesis takes place in the liver. So that is about where it will take place and why it takes place, or what is the importance of gluconeogenesis. Now we'll talk about the exact reaction or what happens in the gluconeogenesis. As I already told, in gluconeogenesis there is a synthesis of new glucose from non-carbohydrate precursor, mainly from the pyruvate. So this pyruvate may be as a result of glycolysis, or it may be produced from the lactate or glycerol, or even from the amino acid. Anyhow, this pyruvate produces new glucose in gluconeogenesis. So it seems to be reverse of glycolysis process, isn't it? Because in glycolysis, there is a production of 2 molecules of pyruvate from the glucose, or 1 molecule of glucose broken down into 2 molecules of pyruvate in glycolysis. Whereas in the case of gluconeogenesis, 2 molecules of pyruvate gives rise to 1 molecule of glucose. So it looks like opposite or reverse reaction of glycolysis, but it is not the case. So gluconeogenesis is not exact reverse reaction of glycolysis, because in glycolysis, that is during the conversion of glucose into pyruvate, some of the reactions are not reversible reaction, or they are irreversible reaction, like production of glucose 6-phosphate from the glucose, production of fructose 1,6-bisphosphate from fructose 6-phosphate, or the last step, that is production of pyruvate from phosphoenol pyruvate. These 3 reactions are irreversible reaction. Irreversible means they cannot take place in the reverse direction. So what happens in gluconeogenesis? Production of glucose from 2 molecules of pyruvate. So reaction should take place in the reverse direction. But in these irreversible step, they cannot take place in the reverse direction. That's why gluconeogenesis is not exact reverse of glycolysis, or it is not the exact opposite reaction of glycolysis. So there is a change in the gluconeogenesis and glycolysis step. So we'll study the changes or differences between the glycolysis and the gluconeogenesis while studying the each steps of gluconeogenesis. In the first step, as I already told, the first step is from the pyruvate. So glucose production starts from the 2 molecule of pyruvate. This pyruvate may be produced from the lactate, glycerol, or any other non-carbohydrate precursor. So from 2 pyruvate, there is a production of oxaloacetate, because pyruvate cannot convert it into phosphoenol pyruvate directly, because it is an irreversible reaction. So this gluconeogenesis bypass this reaction or skip this step by converting it into oxaloacetate. So first the pyruvate is converted into oxaloacetate with the help of enzyme known as a pyruvate carboxylase. These reactions look simple as it is written in the board, but conversion of pyruvate to oxaloacetate is a complex reaction which consisting of many reactions. At first, the two molecules of pyruvate which are present in the cytoplasm, it may be produced as a result of glycolysis or maybe from the lactate or from the glycerol or from any other reaction. These two molecules of pyruvate which are produced in the cytosol enters mitochondria. Later in the mitochondria, these two molecules of pyruvate is converted into oxaloacetate in the presence of enzyme known as pyruvate carboxylase with the presence or with the help of ATP, biotin and carbonic acid. So pyruvate in the mitochondria is converted into oxaloacetate with the help of enzyme known as pyruvate carboxylase in the presence of ATP and biotin and carbonic acid. So pyruvate is converted into oxaloacetate in mitochondria. Later, this oxaloacetate cannot exit mitochondria. That's why oxaloacetate is converted into malate in the presence of enzyme known as malate dehydrogenase, during which two molecule of NADH is oxidized to NAD+. Later, this malate in mitochondria can exit mitochondria. So, malate exit mitochondria and enter cytosol or cytoplasm. And this malate in the cytoplasm is converted into oxaloacetate in the presence of enzyme known as malate dehydrogenase, during which two molecule of NAD+ is reduced to two molecule of NADH. Now, this oxaloacetate in the cytoplasm is converted into phosphoenol pyruvate in the presence of enzyme known as phosphoenol pyruvate carboxykinase or PEPCK with the help of two GTP molecules. So, GTP is converted into GDP, during which oxaloacetate in the cytoplasm is converted into phosphoenol pyruvate in the presence of enzyme known as PEPCK. Now, this phosphoenol pyruvate is converted into 2-phosphoglycerate in the presence of enzyme known as enolase. 2-phosphoglycerate later converted into 3-phosphoglycerate in the presence of enzyme known as phosphoglycerate mutase. In the next step, 3-phosphoglycerate is converted into 1,3-bisphosphoglycerate in the presence of enzyme known as phosphoglycerate kinase. Later, 1,3-bisphosphoglycerate is converted into glyceraldehyde 3-phosphate in the presence of enzyme known as glyceraldehyde phosphate dehydrogenase. In the next step, this glyceraldehyde 3-phosphate is converted into fructose 1,6-bisphosphate in the presence of enzyme known as aldolase. So, all these reactions are similar to that of glycolysis but takes place in a reverse direction because all these reactions are a reversible reaction. That's why it is similar to that of glycolysis but takes place in a reverse direction. In the next step, fructose 1,6-bisphosphate need to be converted into fructose 6-phosphate, but it is not easy because this step is a reversible step. But we have another enzyme which comes into picture in this step, that is fructose 1,6-bisphosphatase. So, fructose 1,6-bisphosphate is converted into fructose 6-phosphate in the presence of enzyme known as fructose 1,6-bisphosphatase with the release of inorganic phosphate. In the next step, this fructose 6-phosphate is converted into glucose 6-phosphate in the presence of enzyme known as phosphohexase isomerase. In the next step, glucose 6-phosphate need to be converted into glucose, which is a last step of gluconeogenesis. But it is not easy again because this step is a reversible step. So, glucose 6-phosphate is stuck at this step. So, before conversion into glucose, this glucose 6-phosphate enters endoplasmic reticulum. So, it enters a lumen of endoplasmic reticulum where this glucose 6-phosphate is converted into glucose in the presence of enzyme known as glucose 6-phosphatase with the release of inorganic phosphate. So, glucose 6-phosphate enters endoplasmic reticulum lumen where it is converted into glucose or cleaved into glucose and inorganic phosphate in the presence of enzyme known as glucose 6-phosphatase. Later, from the lumen of endoplasmic reticulum, this glucose enters cytosol or cytoplasm and later it will be transported to the target site. So that's how one molecule of glucose is produced from two molecules of pyruvate in the gluconeogenesis process. I hope this video is useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.